I'd like to apologise for John McCafferty not being able to make today's meeting, so I'm stepping in for him, but yeah, hopefully I'll be able to do the talk some justice. So what I'm going to do, I'll give some background to IANSAS, which is a small startup company based in Cambridge, UK. And then I'll talk about the way that we usually actually go through our antibody discovery process, but then I'd like to talk about some new technology we've developed where we're directly screening for antibody function by doing phenotypic selections. And also we're developing a new mammalian antibody display platform, similar to the Adimab platform, where they display antibodies on yeast, but we're developing technology where we're displaying antibodies directly on the surface of mammalian cells and then doing selections in that way. And we think that could have an advantage to screen directly for developability of antibodies. And lastly, I'll touch on some work we've been doing very recently where we've been actually displaying T-cell receptors on the surface of mammalian cells, including Jurkat cells, and screening for function. And we think that this is going to be useful in the whole area of immunomodulation and engineering T-cells for therapy for human cells. So ANSYS started about 3 years ago with just 3 people with a single client project. We've done quite well up to now. So we've actually signed 10 different antibody deals with 6 different companies, 3 US-based, several in the UK, and even a Russian company. And every year our turnover's been doubling. We're making a profit now actually on these deals that we're doing with clients, which started off as a pure fee-for-service contract research organisation type model, but more recently we've started actually doing deals where we're actually getting money where we reach certain milestones in projects, and also we're starting to do deals now where we actually do the antibody discovery in-house and actually license the antibodies to external companies, which is more lucrative than actually doing pure CRO work. So as I was saying, so at the moment we have about 14 people in the company, so it's still quite small, and about 2/3 of the staff are actually doing client-based work, where we're working on many different projects ranging from arthritis, immunology type areas through to cancer, and also we're working on checkpoint inhibitor antibodies as well. But about 1/3 of us are actually developing our own in-house research, and I'd like to talk about that mainly in today's talk. And we filed our first patent that was published at Easter this year on mammalian antibody display. So this is just a slide describing what we do in the process of antibody discovery. And I should say, actually, before we started the company, we were an academic group based at the University of Cambridge, and we used to get Cancer Research UK grants in to do antibody discovery on various targets. And prior to that, we also ran a large-scale proteomics project where we were making antibodies to use in immunohistochemistry. So we've used a lot of the knowledge that we've generated in the last 10 years actually to help found the company. And I should say also that John McCafferty was one of the founders of Cambridge Antibody Technology and an inventor of antibody phage display. So we start with target selection. So we can actually make a lot of the antigens in-house. We go through lead isolation, where we actually have an in-house antibody phage display library, a very large naive library. Go through primary screening. We've usually include not only ELISAs but also cell-based functional assays here, and then we can go through affinity maturation, lead optimization, and secondary screening. And at that point, we normally hand over the project to clients. So we get to the point where we generate a lead antibody and a backup with the properties of the client would like, and then we hand them over, and normally then the company would go on and start doing in vivo animal work. So at the heart of the company is really an extremely good, well-validated antibody phage display library. And this was generated by taking the lymphocytes of 50 different human donors, and we cloned the variable heavy and light chain fragments from that library to make a pre-cloned intermediate library. And from this library, we generated the original McCafferty library, and we published work that we did mainly at the Sanger Institute in 2007 where we describe the generation of antibodies to 400 different targets. So that's the way to really tell if a library is a very good one, is the diversity of antibodies actually that is contained in the library. And in this paper, we were able to show that we were able to isolate very specific antibodies to 400 different targets. And at that point, this was one of the largest studies that had been done on a particular library. So in the company, we've actually improved the original McAfee library. So we've gone back to this original pre-cloned intermediate library and made a library 4 times bigger. And we've developed methods to actually have a higher proportion of inserts in the library. So there was some cDNA contamination, actually non-antibody cDNA, in the original McAfee library, and we've eliminated that now in this library. And also we've enabled it easier to actually convert from single-chain antibody format to IgG format in this library, and we can actually go to several different formats. Okay, so I probably don't need to describe antibody phage display selection to this audience, but very briefly, we start with this library, which is rescued to create filamentous bacteriophage displaying single chains as part of the gene 3 coat protein fusion. And then we go through selections, and we can do this with antigen coated on solid phase. We can also use Barton-lated antigen to do solution phase selections, and then we wash, elute the phage, infect the population of E. coli to expand the first round, and then go round to a second round. And then we usually subclone the single-chain antibody population into a soluble E. coli expression vector. And we prefer to do ELISA screens actually as single chains, because we find that if we do phage ELISAs, we get a very high proportion of positives, but these are usually where they can be low expressing antibodies or antibodies with low affinity. So it's better to actually subclone into monovalent format to actually do the ELISA screening. So this is just a slide illustrating that we have a very good library. So for some client projects now, we've able to, just after 2 rounds of selection, identify antibodies with an affinity of less than 100 picomolar. But again, this varies from project to project. But this is just an illustration of several different targets, where after subcloning around 2 output population, we identify between 16 to 24% positives. And I should say actually that these selections were performed with a guy that came into the lab, and we trained him for about 2 months, and then he did the selections. So normally we identify positives, but probably more in the 30 to 40% hit rate. But the impressive thing is that these hits, when you actually go ahead and sequence them, the number of unique VHCDR3s are in the 46% to 93% rate. And if you look at the unique light and heavy chain CDR3 combinations, these percentages are even higher. So this is very important when you're actually going through a therapeutic program, because you want to cover as much epitope diversity as you can when you're trying to identify antibodies that will block, for example, a receptor-ligand interaction or have a particular property. So that's the ability to isolate many different antibodies with high sequence diversity is crucial in the whole pipelines that we run. Okay, so I've gone through how we would actually go through an antibody discovery project with standard projects, but we started actually when we were academics to think of ways to actually screen for function directly, and this could be useful if for example, the target is a difficult target, such as a GPCR or ion channel, or if the receptor actually exists as part of a multi-protein complex, where it's difficult to actually express a particular antigen and have the same conformation as would exist in an in vivo situation. So in grey, this is the standard way that we actually progress through discovery projects for client projects. So we subclone output populations from phage, pick thousands of clones. So this is normally 96 well plates of clones. We perform ELISA. At this stage, we normally also check for species cross-reactivity. So for many client projects, we need to check if they're mouse and monkey cross-reactive, and also that they don't cross-react to a homologue. in human. And then we select unique binding clones, so we actually sequence these. And then we reclone and express the antibodies in mammalian cells, because we usually don't actually perform in cell-based functional screening in mammalian cells with single-chain antibodies expressed in E. coli because of endotoxin contamination. So it's better to actually get them to IgG format at this point. And then we express, purify, concentrate and quantitate the antibodies, test in cell-based assays, and then identify functional antibodies in that process. So this whole process is quite long and involved. Normally we wouldn't be able to do that in less than 4 to 6 months from the start of a project. But more recently, we've started actually taking a whole round to output population, and we have a trick to actually to keep the linkage of the heavy and light chains from single-chain format and clone these into an IgG formatted vector, and then start doing the screening. More people now are actually, so Mammalian are now doing a lot more work where they're screening directly as IgG format. So to actually do a phenotypic screen, what this requires is actually to take the antibody genes and then introduce them into mammalian cells so that there's only 1 or 2 antibody genes per cell, and then perform a phenotypic selection. And in this way, you shortcut this whole process and identify a functional antibody straight away. So we started looking at embryonic stem cells as a generic reporter. So stem cells are interesting cells because they're poised to differentiate into many different subtypes. So you can see that this would be a pluripotent stem cell that could differentiate into a precursor cell that would then differentiate into daughter cells. Now you can imagine that if you're able to block step A, then you would maintain pluripotency. Alternatively, if you blocked a downstream step in differentiation, then you would direct the cells to a different outcome. So to illustrate this process, we actually decided to focus on the FGFR pathway, which is known to be important in maintaining pluripotency in stem cells. So we created a targeting vector with long homology arms, so 3.3 kb on the left arm, 9.2 kb on the right arm, homologous to the ROSA 26 locus. So this is a locus commonly used in mouse stem cell biology because it's expressed in many different cell types. So we had this large targeting vector. So the targeting vector was about 20 kilobases in total, and then we had this TET on promoter, and then we introduced antibody genes. So these are single-chain FC genes flanked by gateway sites. So we could actually take a round-to-output population, convert that to an entry plasmid, and then do a LR recombination into this targeting vector. And we could show that we could actually introduce antibody genes into stem cells, and this is just an example where we're able to show expression of a single-chain FC, where in the absence of doxycycline, there's no antibody expressed into the supernascent of the stem cells, whereas in the presence of 1 or 2 micrograms doxycycline, you can see the single-chain FC antibody being expressed. So in this proof of concept experiment, we took round-to-output populations where antibodies had been selected either on FGF4 or FGFR1 or FGFR2, and then cloned these antibody populations into the targeting vector, and then transformed stem cells and selected for puromycin resistance. And then we took these after 2 days of puromycin selection, we then placed the cells into semi-solid media in differentiation conditions. So this is actually in differentiation conditions that promote neural differentiation. But we actually used also cells that had OCT4 promoter upstream of a GFP reporter. So if cells maintain their pluripotency, then you would expect to see green colonies appearing in this agar plate, whereas the differentiated cells would be GFP negative, and they would also start to lose their compact shape. Sorry. So we published this work in PNS about 2 years ago, and we were able to actually basically identify blocking antibodies of the FGFR signaling pathways by picking the green colonies, isolating the antibodies back from that have been integrated into the genomic DNA, expressing those antibodies, and then showing that we could add the purified antibodies back to stem cells and show that we could maintain pluripotency. So it was proof of concept to show that we could actually go through this phenotypic selection screening and identify antibodies that were functionally active in this particular system. Now, for the work that we're now doing in the company, what we'd really like to do is make much larger libraries. And the problem with this particular method is that it used homologous recombination, which isn't a very efficient way to actually perform gene integration. So we were really only, after puromycin selection and transfection with micrograms of DNA, we were only identifying hundreds to 1,000 colonies. So if you think of how many antibodies are screened in a normal antibody phage display selection, we're looking at library sizes of 10 to the 10. It's not really efficient enough recombination system for us to be able to create large antibody libraries. Yes, which brings me on to the next pass of the talk, which is how do you screen tens of millions of antibody clones integrated into mammalian cells, and also we need 1 or 2 genes per cell. Now, the formats I've been talking about then are single chains that express in E. coli, but we can also screen as single chain FC or IgG or Fab. Yeah, actually, so this is going into a developability talk. And again, I think some of the speakers later actually will go into this. But we're really looking for a sweet spot when we perform antibody discovery, where we have a combination of affinity, specificity, epitope function, and well-behaved. And we think that what we're developing in the mammalian display system will actually enable us to get quicker to a developable antibody. Now, the solution that we came up with for efficient integration of antibody genes into mammalian cells was actually nuclease-directed integration. So this is where you actually would cut the genome in a specific location, and then that enables you to increase the efficiency of homologous recombination by more than 40,000 fold. Now, there are several different ways in which you can actually introduce a specific cut. So one of these would be using a meganuclease, which specifically recognizes an 18 base pair recognition site. However, you would need to introduce that recognition site into the genome, which could be performed by homologous recombination. And we've actually produced cell lines already in HEC cells and CHO cells where we have meganuclease sites introduced. So meganucleases were described about 20 years ago. Zinc finger nucleases were being described probably about 10 years ago. So this again is starting to address the idea that you could actually design zinc finger nucleases to actually recognize specific genomic sections and actually cut them in very specific locations. But tail nucleases are probably starting to supersede zinc finger nucleases because they're, if anything, they're simpler to actually design tail nucleases than zinc finger nucleases. And this shows a tail nuclease actually recognizing a double-stranded DNA helix. Now, probably a couple of years ago, the CRISPR-Cas9 system has come out, and this is now probably superseding the tail nuclease system. But from a commercial point of view, it's kind of difficult to see your way through the patent landscape here. There's probably about 3 patents. So although we've done work where we've used CRISPR-Cas9 to actually introduce double-stranded breaks, we're finding it quite difficult to work out if we're able to use that system commercially. But there's no doubt it's a very efficient way to perform gene integration. Okay, so in the last part of my talk, what I'd like to do is just go through some of the data that we've already generated where we've introduced antibody genes into mammalian cells. And a lot of the work that we've done so far is using the tail nuclease approach. And this is where we create a targeting vector with homologous arms with a left and a right arm homology arm. And here we're targeting the AAVS locus. And again, this is a housekeeping locus expressed in many different cell types. And the AAVS actually has a promoter that drives a phosphatase. And what we do here is we're integrating antibody genes actually into the AAVS locus, but we're relying on the promoter within the AAVS locus to drive antibody expression. So we use tail nuclease to make a double-strand break in the genome, and then we transfect with our targeting vector, which then integrates the antibody cassette into the AAVS locus. And we actually have a blastocidin gene that has no promoter, but the expression is driven by the AAVS locus. And this is to stop non-specific integration, because obviously there are always backgrounds of non-specific integration of targeting cassettes into mammalian cells, and we want to try and select away for the non-specific integration, because here we are trying to get 1 or 2 copies of our antibody gene per mammalian cell. So we should theoretically only see a blastocidin-resistant colony if the integration has gone into the right locus with an upstream promoter. So downstream of the blastocidin cassette, we have a PEF promoter driving the light chain, CMV promoter driving the heavy chain to produce the antibody cassette. So in our initial experiments, we could show that we could do the transfection to select for blastocidin-resistant colonies, and then actually show that we were seeing expression of the antibody on the surface. And actually in this vector map, I should say, there's actually a transmembrane domain downstream of the heavy chain to enable membrane tethering of the antibody. So we wanted to show that we were getting 1 antibody gene per cell, which is important for the selection. And here what we did was we transfected with a 50-50 mix of an anti-FGFR1 and anti-FGFR2 antibody, and then showed by flow cytometry the proportion of either single positive for FGFR1 or 2, or double positive in this flow cytometry diagram. And the proportion of double positive is related to the amount of DNA that actually is used for the transfection. So here we actually had a 1-to-1 mix of the donor DNA encoding the antibodies with the tail nuclease, but here we had an excess of the antibody gene. You can see that we're getting more of a double positive population. Okay, so that was showing single chain FC display on the surface of mammalian cells. We've also shown that we can display full IgGs on the surface of mammalian cells. So here we took a round 2 output of antibody selected on beta-galactosidin, and again went through the selection process, and showed that we could see the specific display of anti-beta-galactosidase antibodies. So here we took the output of a first round selection. So of the unsorted cells, 3.2% were positive for beta-galactosidase and for FC expression. But after selection, so this is where we maintain the cells in the presence of blastocidin, and you can see that 62% of the cells actually display antibody on the surface. Actually, in the interest of time, I'm going to skip through this slide. Yeah, this is more of interest for the hardcore phage display guys, I think, because yeah, I won't talk about this at all. This slide is interesting, actually, and you see some of the yeast display people talking about this, that depending on how you actually do a sourcing experiment, you can actually select for higher affinity antibodies. So here, when you do a flow sourcing experiment, you can actually choose the concentration of the antigen used in the salt. So here we've used 0.1 nanomolar, 1 nanomolar, or 10 nanomolar, and you can see the number of positive cells appearing in the flow diagram is greater at the higher concentration, and this is a reflection of the range of affinities of the antibodies that are actually being displayed on the surface of mammalian cells. And this is very useful to actually rapidly select for high affinity antibodies. And on this surface, you can actually, this is the concentration of the antigen, but also on the y-axis, you can select for the just with an anti-human FC antibody, the display level as well. So in a flow separation, we're selecting both for the affinity of the antibody and also its expression level on the surface of mammalian cells, which comes back to this idea of trying to select for antibodies with good developability. Okay, so that's all I'm going to talk about regarding the display of antibodies on the surface of mammalian cells. So we think that this has potential actually to be a new technology platform that can either be used in conjunction with antibody phage display, where we clone round to outputs and then do selections, or we're in the process of actually trying to make much larger libraries on the surface of mammalian cells. But for that, we're starting to see that there are some technology issues revolving around the size of the cells that we're actually using. So Heck and CHO cells actually have quite a wide diameter, and I think anyone that's done tissue culture realizes that if you spin down to 10 to the 6, 10 to the 7 cells, you see quite a large cell pellet in the surface, whereas we're moving now to using smaller cells, actually, which could be easier to use in selection experiments. Okay, so just 2 more slides now I'd like to go through. So we've gone through methods to actually display antibodies on the surface of mammalian cells, but another application of performing high efficiency gene integration is actually in the area of displaying T-cell receptors. So T-cell receptors are interesting. They consist of an alpha and beta chain, and this is a structure of a T-cell receptor, which is almost similar to an antibody, but you can see here that it's in complex with an MHC molecule that actually has a peptide embedded in the surface. So you can see that the variable loops are contacting both the MHC molecule and the specific peptide. So if we were able to come up with a way to actually display T-cell receptors and perform selections, we think it would be quite a rapid way to actually identify new T-cell receptor therapies, in the same way that Immunocore has developed a system with their IMTAC system, where they've actually made single-chain constructs of T-cell receptors. We think that if we're able to display on the surface of mammalian cells whole T-cell receptors, we'll be able to select for T-cell therapeutics, either as purified T-cell receptors or even actually integration of T-cell receptors in human white blood cells for cell therapy. So as a proof of concept, we took 2 T-cell receptors that recognize specific cancer epitopes. So we've called these either TCR1 or TCR2. So there's no cross-reactivity between these 2 T-cell receptors. They're highly specific for those particular peptides embedded in MHC molecules. And you can actually purchase these peptides pre-complexed tetrameric MHC from a company in Oxford, and you use these peptide MHC molecules in flow cytometry experiments. So again, we've constructed a targeting vector with AAVS homology arms, We have two promoters driving either the alpha or beta antibody TCR fragments. And we went through the process of doing a transfection and selecting for, in this case, puromycin resistance. And you can see that for labeled peptide 1, we only see binding of MHC, of the relevant MHC to the surface of cells transfected with TCR1, but not TCR2. And for MHC peptide 2, we only see labeling of cells transfected with TCR2, but not TCR1. So we've shown that we can see specificity on the surface of HEC cells here. And also we've done some experiments where we've shown enrichment of where we've done transfections of 100-to-1 ratios of either TCR1 or TCR2 populations, selected on puromycin, and then sorted the cells, and we can see an enrichment of the cells after sourcing on TCR1 or TCR2. So that it gives an indication that we can actually take whole libraries of T-cell receptors and sort them. We've also made a small library of T-cell receptors and also gone through a sourcing process. And this is the last slide I showed. This is where we've actually taken the T-cell receptor, and instead of transfecting HEC or CHO cells, we've actually transfected Jurkat cells, which is a transformed T-cell line. And we've shown that the actual tetrameric MHC molecule can actually activate the Jurkat cells, both in the absence of CD28 or the presence of CD28. And this is shown by the upregulation of CD69, which is a T-cell receptor marker. So this opens up the possibility of actually not just selecting T-cell receptors on the basis of binding, but also selecting for T-cell receptors on the basis of function as well. So we're quite excited by this result. That we generated this only about 3 months ago. Okay, so I'm going to draw this to a close now. So hopefully I've actually gone through some of the technology developments that were required for mammalian cell surface display of antibodies, and this really revolved around making very specific cuts in the genome, and then a more efficient method of antibody or TCR gene integration. And we're hoping that this is going to be a method to screen for tens of millions of antibodies. And then lastly, I've gone through the display of T-cell receptors and binding and activation. Okay, I'd like to thank John McCafferty, who headed the lab at University of Cambridge, and then is now acting as CEO and CSO of Iontas. Anna did a lot of the early work on the stem cell project that I discussed, and Kosei has done a lot of work on the mammalian display system. Thank you very much for your attention.
Thanks, Mike. We have time because of the entire day today. We do have times for questions, even if we go over the individual speakers. So please, if anyone has any questions, it's hard to know where to start. He presented so much between his proprietary technology, the whole idea of a mammalian-based library, and then even including now T-cell receptors. But feel free if anyone has any questions. Sure.
You mentioned earlier that you would use cell-based assays to measure functionality. How is this dependent on the type of cells that you use? What kind is dependent on relevance on the type of cells that you use if you would extrapolate the functionality out of cells?
Yeah, it's difficult actually, because the trouble is that each project is a completely new area almost. A lot of the functional assays we've developed for the client projects are completely different. I think it's one of the key things actually in starting the project that.
But that would depend on the antigen you use. You mentioned that you use cell-based assay across to measure functionality, and I just wondered where sometimes is that how relevant it is sometimes you might get whether you have a good extrapolation measure functionality in larger volume?
Yeah, I think it's difficult. It's something that we've wrestled with actually after the stem cell papers. So how can we make this more generic? And it was actually one of the referees' comments as well. And yeah, it's not an easy problem actually because to make it very generic. Yeah. I think the best that can be done probably is in screening phenotypically is to try and address a particular area perhaps. And, you can imagine in checkpoint inhibitor area perhaps you could think of perhaps a generic phenotypic screen. But I think it's got to be done subject by subject.
Any other questions? Don't be shy. We've got this opportunity. Please take it. We have the time. I have one that's a bit naive. I was first introduced to generating phage libraries maybe 30 years ago, and I've evolved into other areas of study. So I'm just bringing it back now for your talk. But in generating your T cell libraries, my understanding was that the various T cell the variants and the receptors were kind of randomly generated in the body, and so that when a T cell with the proper receptor hit something, it would then divide, it would stimulate, and that's how you would generate this population of the appropriate receptor. So I'm thinking back, where do you get the variants to build your library from? Where do you get this repertoire of possibilities to put into a mammalian cell?
Yeah, it's good. So we are starting to think about making. We haven't started making a naive TCR library yet, but we may go back to the same lymphocytes we used to clone our heavy and light chains for the antibody library, actually, because there will be a population of different TCRs, variable fragments actually within those lymphocytes. But I think we need to use a naive population so that it's not biased in any particular area.
Can you use like Turgid as your base to clone into?
Yes, I think we probably will do that. Yeah, we've yeah.
Anyone else? Okay, well, thanks, Michael. It was very